morning, dear students. In the previous class, we have discussed about the enzymes. We said that enzymes have an active site, a site where which the substrate is going to attach to the enzyme. The most important key point that was discussed in the previous lecture was about the activation energy. We said that activation energy is required for any chemical reaction to proceed in our body. A minimum amount of energy is definitely required, and therefore the substrate is always at a higher energy level compared to the product, and thus the reaction is going to be an exothermic reaction. But whether the substrate is at higher energy level, in order to form the intermediate stages, transient stages, some amount of energy will definitely be required. This energy is not required for the formation of the product, but it is required for the formation of the intermediate products. And thus, this energy, that is the activation energy which is required, can be lowered down in presence of the enzyme. Every reaction will require energy, but the amount of energy required for every reaction can be reduced in presence of the enzymes, and thus enzymes are responsible for fastening the rate of the reaction, that is, increase the rate of the reaction. Now we are moving on to the next topic we need to understand about enzymes, and that is nature of enzyme action. Nature of enzyme action. Now when we are talking about the nature of enzyme action, we know one thing, that the enzymes, all enzymes have an active site. They have an active site for attachment of the substrate. All enzymes will have an active site, and this is the site where the substrate is going to attach to the enzyme. And there is formation of, resulting into formation of enzyme substrate complex. What is going to happen? As soon as the substrate attaches to the active site of the enzyme, the enzyme will properly fit along with the substrate, and thus resulting into formation of the enzyme substrate complex. Now this enzyme substrate complex is highly reactive, and therefore it will change into enzyme product complex. Changes into enzyme product complex. The newly formed product will not be now further able to attach to the active site of the enzyme. एक बार जो प्रोडक्ट बन चुकी है, अब ये प्रोडक्ट जो है, it cannot remain attached to that active site of the enzyme. Therefore, the product gets separated, and we again get the enzyme in the original form. So, the product gets separated and the enzyme is obtained in its original form. The enzyme will be obtained in its original form. So here, this type of nature is the enzyme action. Now if we study the catalytic role of each step, if we study each step in detail, what will be the steps in which this reaction is going to occur? First of all, we are mentioning over here, enzyme combines with the substrate to form enzyme substrate complex, which further forms enzyme product complex, and this gets converted into enzyme plus product. मतलब substrate जो है वो एंजाइम के एक्टिव साइट पे अटैच होता है, फिर एंजाइम सबस्ट्रेट कॉम्प्लेक्स बन जाता है। एंजाइम सबस्ट्रेट कॉम्प्लेक्स चेंज होके the substrate changes into the product, but the product is still attached to the enzyme. Again, this is an intermediate transient unstable stage, and then after it gets separated to enzyme and the product. So what are the steps that are going to happen over here? The first step is the attachment of substrate to the active site of enzyme. First of all, the substrate is going to find out the active site of the enzyme, because active site of the enzyme is having a tertiary structure of protein. Okay, tertiary structure of protein will be forming this active site, and at this active site the substrate is going to attach. The second step that is going to happen is the substrate is going to fit properly, or the enzyme is going to alter its shape in such a way that it fits more properly to the substrate. So enzyme alters its shape in order to properly attach with the substrate. Properly attach with the substrate, or you can say make a more firm attachment, right? पहले क्या होता है कि सबस्ट्रेट जब एंजाइम के एक्टिव साइट पे अटैच हुआ है तो ये लूजली अटैच्ड है। लेटर ऑन क्या होता है कि एंजाइम जो है इट विल बी ऑल्टरिंग इट्स शेप, ऑल्टरिंग इट्स कन्फर्मेशन इन सच अ वे दैट द सबस्ट्रेट कैन मोर प्रॉपरली अटैच विद द एंजाइम. So now there is a firm connection between the enzyme and the substrate. Immediately there will be certain chemical bonds break, and there is formation of new bonds, and the substrate gets converted to the product. So what has happened now? Substrate was firmly attached, but now certain bonds between the substrate and the active site of the enzyme, certain bonds between the substrate and the active site of the enzyme are going to break, and some new bonds are going to be formed, and therefore changing the substrate into the product. What is going to happen? There will be change from the substrate to the product, but the product is still attached to the enzyme, okay, forming enzyme product complex. And lastly, the product gets separated from the active site of enzyme, and the enzyme is obtained again free. So enzyme again is going to become free because the product is no longer able to attach to the active site of the enzyme, as its conformation has changed. So this product is going to be separated out from the enzyme, and at the end of the reaction we again get our enzyme free. We get our enzyme free means now the enzyme is free to again perform the new chemical reaction. So in this way there will be the role of the enzyme action, the nature of the enzyme action. Next we will discuss about some factors which are affecting the enzyme activity. So after discussing about the nature of enzyme action, let us have a look upon the factors affecting the enzyme activity. Now, the enzyme activity is often going to be affected by different factors like temperature, pH as well as substrate concentration. Why we are mentioning that these factors will be affecting the enzyme activity? Because as we know that enzymes are proteins and these are the tertiary structure of the proteins, any change in the temperature or any change in the pH might result into altering the conformation of the proteins. मतलब टेम्परेचर अगर बहुत ही ज़्यादा चेंज हो रहा है या पीएच चेंज हो रहा है तो एंजाइम का जो टर्शियरी स्ट्रक्चर होता है वो डीनेचर हो जाता है और डीनेचर हो जाने की वजह से it doesn't have that proper conformation for attachment to the substrate and therefore the activity of the enzyme is going to be lost due to such altering conditions. So as we know that proteins जब होते हैं और उनमें अगर कोई changes आ जाए जैसे कि temperature या pH में तो proteins उसी तरह functional नहीं रहते और वो denature हो जाते हैं, break हो जाते हैं और सारे ही enzymes क्या है proteins है. So this is going to happen with the enzymes also. So let us have a look upon which factors affect the activity of the enzyme. So first we are going to talk about the factor which is the temperature. So temperature, with the increase in temperature, the activity of enzyme is going to increase. मतलब टेम्परेचर बढ़ने पर एंजाइम की एक्टिविटी भी जो है वो इंक्रीज हो जाती है पर ओनली अप टू सर्टेन एक्सटेंट. ओके, ओनली अप टू सर्टेन लिमिट. ये लिमिट क्या होगी? तो अगर हम ह्यूमंस और सारे एनिमल्स की बात कर रहे हैं तो ये लिमिट होगी नियरली 40 डिग्री सेल्सियस. 40 degrees Celsius is a limit for humans and other animals because we maintain this ambient body temperature throughout our life. There are some organisms which can survive at 80 or 90 degrees Celsius, but they are very few and their enzymes can function even at very high temperature. But for most of the organisms existing on this planet, there is a limit of temperature up to which the enzyme activity is going to increase. After which, if you keep on increasing the temperature, the activity of the enzyme is going to decrease. Okay, a further increase in temperature results into denaturation of the enzyme. It results into denaturation of the enzyme. The temperature up to which it is properly functioning is known as the optimum temperature. मतलब जिस टेम्परेचर तक वो मैक्सिमम काम कर रहा है उसको क्या कहेंगे? जैसे कि 37-38 डिग्री पर एंजाइम परफेक्टली काम करता है विद देयर मैक्सिमम एफर्ट, तो उतने टेम्परेचर को हम क्या कहेंगे? ऑप्टिमम टेम्परेचर कहेंगे. अगर ऑप्टिमम टेम्परेचर से टेम्परेचर बढ़ता गया 45 डिग्रीज 50 डिग्रीज तो एंजाइम एक्टिविटी क्या होने लगेगी? धीरे धीरे कम होने लगेगी एंड एट अ सर्टेन टेम्परेचर द एंजाइम विल कंप्लीटली डीनेचर एंड देयरफॉर इट विल नॉट बी एबल टू परफॉर्म इट्स फंक्शन. एट लोअर टेम्परेचर will make enzyme inactive. मतलब अगर टेम्परेचर बढ़ा दिया है तो एंजाइम डीनेचर हो जाएगा, राइट? राइज इन टेम्परेचर तो एंजाइम डीनेचर हो जाएगा. पर अगर टेम्परेचर बहुत ही कम कर दिया है, वेरी लो टेम्परेचर, then what is going to happen? The enzyme is going to be inactive. Sometimes for certain experiments experiments which are to be performed at other places, एंजाइम जो है उसका फंक्शनिंग लूज ना हो, इसीलिए एंजाइम को लो टेम्परेचर पे रेफ्रिजरेट करके स्टोर भी किया जाता है, which can be made active again on providing ambient temperature. So sometimes enzymes can be stored also and their storage requires low temperature. So how the graph is going to be? If we draw a graph of the temperature versus the progress of reaction what are we going to get? That initially the enzyme action is going to increase with the increase in temperature, but after a certain limit what is going to happen? The activity is going to decrease. The same stands for temperature also as well as for the pH. Enzymes are functional at a normal temperature range. Same way, they are functional even at a specific pH range. Okay. So next factor which is going to affect is pH. Enzymes are active. Enzymes are active at optimum pH रेंज. हर एक एंजाइम जरूरी नहीं है कि एसिडिक पीएच में ही एक्टिवेट हो या बेसिक में. कोई एंजाइम्स ऐसे भी होते हैं जैसे कि हमारे स्टमक में जो पेप्सिन एंजाइम है वो एसिडिक पीएच पर ही काम करता है, और सिमिलरली जो पैंक्रियाटिक अमाइलेज है वो अल्कलाइन पीएच पर ही काम करता है, राइट. तो पैंक्रियाटिक अमाइलेज अल्कलाइन पीएच के लिए उसका ऑप्टिमम पीएच अलग होगा, और कोई एंजाइम अगर स्टमक में है, पेप्सिन है, तो उसका पीएच रेंज अलग होगा. पर कहने का मतलब ये है कि हर एक एंजाइम की खुद की एक स्पेसिफिक पीएच रेंज होती है. उसी पीएच रेंज में वो फंक्शनल होता है. अगर आप उसके पीएच को ऑल्टर कर दोगे, उसके मीडियम के पीएच को ऑल्टर कर दोगे, तो रिएक्शन उस सक्सेसफुली होगा नहीं. Maybe it can happen that reverse reaction might start if you alter the pH condition. So graph of pH is also going to remain same. With the increase in the pH, activity is going to increase. But after an optimum pH, उसका optimum pH 8 हो, 7 हो, कुछ भी हो, पर एक optimum pH के बाद अगर आप pH fluctuate करते हो तो enzyme action क्या हो जाएगा? Decrease हो जाएगा. Okay. The third factor that affects is the substrate concentration. The third factor which is very important is the substrate concentration. So here also increase in substrate concentration increases the rate of reaction, right? But again up to certain limit, up to certain extent. Why certain extent? Because beyond this extent, if you keep on increasing the substrate concentration, the number of enzyme molecules are few. सारे ही enzymes पे एक substrate तो attach हो ही गया है. So now the enzymes have got saturated with the substrate. So after saturation, if you keep on adding substrate in the medium, the enzyme is not going to increase the rate of the reaction, because at a particular concentration all active sites of enzymes are saturated with substrate, right? At a particular concentration, substrate का concentration है, तो मान लो 10 एंजाइम मॉलिक्यूल्स हैं, तो उनके साथ कितने सबस्ट्रेट अटैच हो सकते हैं? 10 ही हो सकते हैं. फिर आप मीडियम में 20, 30, 40 सबस्ट्रेट ऐड करते रहो, पर एंजाइम तो 10 ही है. सो दे आर नॉट गोइंग टू फर्दर इंक्रीज द रेट ऑफ द रिएक्शन, बिकॉज द एंजाइम्स एक्टिव साइट्स आर नाउ ऑक्युपाइड बाय ऑल द सबस्ट्रेट मॉलिक्यूल्स. देयर आर नो मोर एंजाइम्स टू अटैच विद द substrate. So even if you keep on increasing the substrate concentration, it is not going to make rate of the reaction, and this is represented as, right? देखिए अब क्या हुआ होगा, that at a particular concentration, इसको मान लेते हैं substrate concentration, इसको मान लेते हैं progress of reaction या rate of reaction is going to progress in this direction, ठीक है। तो यहाँ पे क्या हुआ कि जैसे जैसे हम सबस्ट्रेट का अमाउंट बढ़ाते गए वैसे वैसे रेट ऑफ रिएक्शन जो है वो बढ़ता गया, पर एक लिमिट आ गई है और इस लिमिट के बाद इस लिमिट को कहा जाता है Vmax। व्हाट इट इज नोन एज Vmax। Vmax मतलब a substrate concentration at which the rate of reaction is maximum। a substrate concentration at which the rate of the reaction has become maximum, increase in substrate concentration will now no longer result into increase in the reaction, okay, that is known as Vmax। now another important term that is mentioned in your textbook is a word known as an alphabet Km। अगर आप आपकी टेक्स्ट बुक में देखोगे तो ऐसे ही ग्राफ के नीचे यहाँ पे लिखा है Km, which is known as Michaelis-Menten constant। Michaelis-Menten constant, जिसका मतलब ये है the concentration of the substrate at which the rate of reaction is half of its maximum। the substrate concentration at which the rate of the reaction is half of its maximum। rate of reaction जो है वो maximum से कितना हो गया, हाफ। तो कितने substrate concentration पे reaction maximum का हाफ है, right? that half Vmax is the denoted as the Km। Km value मतलब it is the amount at which the rate of the reaction has become half of its maximum velocity, ठीक है। तो यहाँ पे Km Vmax बहुत ही important है, right? so factors which are affecting the enzyme activity में temperature बढ़ता जाएगा, enzyme activity बढ़ती जाएगी up to an optimum temperature। same way enzymes are active at a specific pH। if you further change the pH, the activity of enzyme will decrease। with the increase in substrate concentration, rate of reaction is going to increase again up to a certain limit, after which if you keep on increasing the amount of substrate, the rate of reaction is not going to change, it is not going to further increase। so these are three factors। इसके अलावा in the next topic we will discuss about enzyme inhibition, which is also considered as a factor which affects enzyme activity। so we are going to talk about enzyme inhibition and nomenclature of the enzyme।